Μπορώ να έχω την πρώτη διαφάνεια? Ωραία, οκ. Προχωράμε στην τελευταία συνεδρίαση για απόψε. I have the honor to present Professor Katarzyna Bogunia-Kubik, who is a distinguished scientist with expertise in human immunology and immunogenetics, combining research and diagnostics. Όχι, συγγνώμη, μην το παίρνετε. Ωραία. And focusing on histocompatibility testing for organ transplantation, genetic predisposition for autoimmune disease, and diagnosis of infertility and recurrent pregnancy loss. Dr. Bogunia is the head of the Laboratory of Clinical Immunogenetics and Pharmacogenetics in Hirschfeld Institute of Immunology and Experimental Therapy in Polish Academy of Science in Wrocław, Poland. She is the co-authored in 10 books, chapters, and over 160 scientific publications. And she is currently a member of the Scientific Council of Hirschfeld Institute, of National Institute of Geriatric Rheumatology and Rehabilitation, on the European Board of Transplant Immunology, and in the Board of Polish Society for Immunogenetics, past president and honorary member. But I would like to mention that in her involvement in European Federation for Immunogenetics, as she was past EFI Executive Committee Councillor and currently member of Educational and EPT Committee. And here in the two photos, you can see the current EPT and Educational Committee. I had the opportunity to know her better during our joint participation in EFI Executive Committee as councillors in 2019. Since then, a collaboration between the two labs in Hirschfeld Institute and Evangelismos Hospital was started. As a result, several presentations in scientific meetings as well publications were produced. Additionally, young scientists from Poland came to our lab in Evangelismos Hospital for short-term training. Now, I would ask from my co-chair, Dr. Chryssia Papasteriadis, to give a short introduction. I would like to mention, especially for the young members of our society, that Dr. Papasteriadis, as director of Immunology Histocompatibility Lab of Evangelismos Hospital, established our department as reference center for disease immunogenetics in 1991, the only existing in Greece. Thank you, Katerina. Έχει ανοιχτό, το έχεις ανοίξει, όχι μάλλον. Κάτω, πιο κάτω, εκεί, μπράβο, εντάξει.
Okay, thank you, Katerina. It is also my great pleasure to welcome Professor Kubik in our conference and in Athens. And Professor Kubik, I am really impressed from your curriculum vitae, so many activities. Since Katerina referred to the laboratory at Evangelismos Hospital and your collaboration, I would like to say that this collaboration started many, many years ago, in 1991 actually, when Dr. Anna Goral from Warsaw was participated in a European multi-center program on SLE immunogenetics. So the two countries had the opportunity to analyze and to compare our results on the genetics of this disease. And I am glad this collaboration goes on nowadays with the two laboratories. I think it is in rheumatoid arthritis patients. Okay, I wish you good results on that. Dear participants, rheumatoid arthritis is an autoimmune inflammatory systemic disease that affects approximately 1% of the general population. Multiple genetic and environmental factors have been linked to either increased or decreased the risk of developing the disease. From an immunogenetic point of view, the MHC region plays the major role, and it is being intensively studied and documented. Nowadays, research is also pointing to additional genetic factors and on the contribution of virus immune cells like NK cells. From that, all these studies has been suggested that NK cells may play both protective and pathogenic roles in rheumatoid arthritis pathogenesis. NK cells can act in these cases both either through their surface molecular proteins, membranes, receptors, or through their cytokines they produce. Professor Kubik has been deeply involved in this line of research, and today she will share with us the current data on the field of in her talk, which is entitled NK Cells Immunogenetics in Rheumatoid Arthritis. Please, Professor, take the floor.
First of all, I would like to thank you very much for this kind invitation to come and participate in such an interesting meeting, although in Greek only. So I would like to thank you, especially Katerina, for this invitation. And I was asked to speak about the immunogenetics of rheumatoid arthritis, but I modified the title a little bit to focus more on NK cells as well. Okay, so as we have already heard from this introduction words, so NK cells can act either by secreting cytokines or chemokines or by releasing apoptotic inducing effector granules. They compose up to 15% of lymphocytes and that have been discovered in 1970s. They play a role first in the line of defense of the innate immune system, as they are essential for protection against viral infection and detection of malignant cells. But what is also important, so they have been implicated in the development of many autoimmune diseases. And among them, we can see Beckett disease, ankylosing spondylitis, psoriasis, multiple sclerosis, systemic lupus erythematosus, autoimmune diabetes, and also rheumatoid arthritis, which we will learn more about today. So NK cells, they are activated by the balance between the activatory and inhibitory receptors. Most of them that have been already very deeply studied belong to the KIR family, but there are also other families of natural cytotoxicity receptors, NCRs, also those associated with NKG2 or immunoglobulin-like transcripts. And these receptors, similarly as KIR receptors, can possess also either inhibitory or activatory activity. And for example, these ILDs, they are inhibitory receptors. NCRs belong to the activation ones, while among this NKG2 family, we can distinguish both NKG2A inhibitory receptors and two other belonging to these activatory ones. So I would like to focus more on this NKG2 receptor, which can form, as you can see, a homodimer. It does not contain this CD90 molecule, and also it does not contain this either I-team or E-team activation or inhibitory motifs, and is associated with DAP10 molecule. It has two ligands, MIC-A and MIC-B. molecule, and this receptor can be expressed on NK cells, but also could be found on CD8 cells, gamma delta T cells, B cells, and the others, and the receptors is encoded within 12 chromosomes. So as I already said, so the signal from this receptor is coming through DAP10 molecule, and it is the activation signal. Coming to the ligand, so it is MIG-A or MIG-Me molecule. As we know, MIG molecules are encoded on the short arm of chromosome 6, similar as other HLA molecules, although MIG-A does not belong to this classical HLA molecules. It looks quite similar to the heavy chain of molecular MHC class 1 molecule, but does not is not associated with beta-2 microglobulin. So as we can see also, it contains very narrow peptide binding groove, and in the consequence is not does not play so important role in the peptide presentation. So in our studies, we have focused on some genetic polymorphisms that are located within MIG-A, but also NKG2D receptor. And as we have heard from the previous speaker, it is important to know what I have understood from this presentation, is that it is important to know how the change within the DNA sequence can affect the protein expression and this protein structure. And for our studies, we have selected such polymorphism that are associated with the changing of amino acid, and this one is quite important because it is associated with the amino acid sequence within the transmembrane domain. This one, which play an important role with this interaction with DAP10 molecule. Another one. This is potential transcription binding site that can have possible impact of NKG2D expression. Another one is also very much is very interesting because it is located with 3' UTR region, and it is associated directly with activity of NK cells. And here MIG-A molecules polymorphisms that have been studied, two of them are missense, as this one is especially interesting because it's associated with the affinity of binding to the receptor. And there have been quite interesting studies showing that dependently of which variant is present on the target cell, so the activity of these NK cells could be changed. If this is valine, so then the degree of NK cell cytotoxicity as well as interferon gamma production increases steadily with the MICA expression on the cells. But in contrast, the augmented expression of this MICA isoform, this one which is associated with high affinity, has none or even a negative effect of NK cell function. But why? This is because of the rapid downregulation of the receptor on NK cells. So we have already heard about this rheumatoid arthritis, this disease usually that develops at the fourth decade of age. Women are affected 3 times more than men, and there are very many risk factors, but among them the genetics play an important role. And now I would like to recall one of our studies performed together with the with the group of Dr. Tarashi from Evangelismos Hospital when we looked at the distribution of HLA DRB1 variants that are characteristic for this rheumatoid epitope sequence. As you can see here, majority of patients, both from Greece and from Poland, they characterized with the presence of this short epitope sequence. Majority of patients in both groups were carrying the DRB1-01 allele. But as we can expect, our population are different, and this is also seen with the distribution of our other alleles in Poland. In Polish patients, we have 0104 and 0401, while in Greeks 0405 and 1001 allele. But as I mentioned before, we also looked at the distribution of NKG2D polymorphism, and please note that this CC homozygosity is less frequent among Greek patients as compared to the Polish patients. But if this genotype is present, the Greek patients characterized with more severe disease, what is shown by the higher disease activity score. Also, there are different associations between MICA variants and the hallmarks of the disease in Polish and Greek patients. So, the presence of rheumatoid epitope, especially DRB1-01, is more frequently seen among Poles, lacking MIC-AGG homozygosity, while among Greek patients, the presence of anti-CCP antibodies and rheumatoid factors is more frequent among those patients who possess this GG homozygous genotype. We looked further among the associations and impact of MIC-A and NKG2 variants on the treatment outcome in the patients that had received anti-TNF therapy, and this was done within our Polish population. This was about 300 patients involved in this study. As we can see here, that GG homozygosity, also here on this graph, is associated with lack of the response among in the patients to the anti-TNF therapy, while those patients who possess heterozygous genotype, the response to the treatment in those heterozygous cases is much more favorable, is good or intermediate. I would like to tell that also this heterozygosity seems to have an important role looking at this NKG2 genetic variants in the same group of patients. Once again, no response was seen in those patients who were here is quite an interesting observation, because in those patients do not respond to the treatment who possess the genotypes that predispose to decrease NK cell activity. So this is either that they have this GG homozygosity here, or they lack this GG variant with respect to this second polymorphism. But the good or intermediate response, once again, is more favorable within the group of patients who were heterozygous. So it means that just the balance between this activatory, that the small balance, but that the medium activation of NK cells is necessary to elicit this more favorable response. Knowing that this NKG2D receptor can play an important role in the patients and its genetic variants, we also looked at the changes of the expression of this receptor on NK cells in patients during the treatment. So it was at the baseline 3 months and 6 months of the treatment, and we compared this with the healthy controls. And as you can see here, the expression of these activatory receptors on NK cells is quite high, but a little bit decreasing, but is much higher, significantly higher at the all time points than those observed in the healthy controls. We also took into consideration another activatory receptor from this second group of natural cytotoxicity receptors, NCR3. And we also observed that the NK cell that majority of NK cells express this activity receptor, but the number of the frequency of the cells decrease during the treatment, and in the case of this NCR3, these differences are also significant. This NCR3 is interesting because when the solute protein is found within the synovial fluid, that usually patients suffer much more with respect to this inflammation of the joints. So we, of course, as immunogenetics, once again looked at the distribution of different NCR3 variants in our patients. So please note that this homozygosity here, AA, seems to play a negative role because we observed in our patients that they develop disease at the younger age, that develop the disease despite also in the absence of rheumatoid factors, and also the response to the treatment seems to be worse because the CRP level after 3 months, 12 weeks of anti-TNF therapy is higher in those patients with this AA homozygosity. Looking at the markers of cytotoxicity, we can also not only look at this receptor that expressed on the cell surface, but also at the molecules that can be released by the NK cells, leading to the apoptosis of the target cells. And I would like now to tell a little bit more about granzyme B and perforin. So we look at the granzyme and perforin levels in the patients and controls, and what we observed that the perforin level decreased during the treatment, while this granzyme level is elevated as compared to controls in patients before the anti-TNF therapy. And once again, we looked also whether we can find any association with the polymorphism that is located within the gene coding for this granzyme B protein. And what we can see here on the right of this slide, which is quite busy, I know, so that the presence of T allele is associated with higher disease activity score in patients both at the baseline and after 3 months of the treatment, while the homozygosity is more frequently present in patients with higher levels of granzyme B before treatment. So coming to the end of my presentation, I would like to show just very recent data, because we also having some DNA samples left in the fridge. We also typed the Greek patients from the polymorphisms located within the granzyme B gene, perforin gene, and also this NCR3 receptor, this activating one. And what we have noticed that Greek patients, less frequently present with this AA homozygous genotype as well as the A allele. And we know that this disease is more frequent among females than males. And we notice also that there are some differences in the distribution of this A allele, which is more present in the female patients. This AA homozygosity is also present more in the female patients, but maybe if you remember, also in the Polish patient, we observed the negative impact of this polymorphism of the onset of the disease. So also here, these Greek patients with this A allele, they develop disease in a younger age. So summarizing, so the risk of the developing rheumatoid arthritis is associated with various HLA, DRB, short epitope alleles in Greeks and Poles. In both populations, genetic variants of MIC-A and NKG2D genes may have disease development, but rather through the different association with the short epitope alleles, anti-CCP antibodies, or rheumatoid factor. MICA genotypes associated with reduced NK cell activity seem to play a negative impact on the efficacy of anti-TNF treatment, whereas MIC-A and NKG2D heterozygosity likely leading to moderate signaling through the NKG2 pathway, which may result in the most appropriate activation of immune effector cells involved in the mechanism of anti-TNF response, is important issue in the context of effective anti-TNF therapy. NK cells seem to be hyperactivated in patients with RA, as increased frequency of NK cells expressing activating receptors was observed. However, the percentage of activated NK cells, especially positive for the NCR3 receptor, as well as perforin secretion, they decrease during the treatment. Prior to anti-TNF treatment, granzyme B levels are higher in patients with RA than in healthy control subjects and are associated with granzyme B gene homozygosity, whereas the disease activity, DAS28, is higher at the baseline and after 3 months of the treatment in patients with the T allele. So, one second. So this NCR3 polymorphism, as we observed in both populations, may affect the disease onset. And in conclusion, what I could say that this observation may suggest that NK cell immunogenetics plays an important role in the pathology of the disease and the efficacy of anti-TNF treatment. And I would like to thank you, our clinicians from the 2 hospitals, from the Medical University of Wrocław, and also from the hospital in Bydgoszcz. I would also like to thank you, our collaboration from Greece, from the Evangelismos Hospital, from the Department of Katerina Terashi, and from the Rheumatology Department as well. And I would like to thank my colleagues that have been involved in this study, especially Milena Iwaszko on the right, and also 2 younger girls. Here we have Sylwia Biały. She just yesterday had the opening of her PhD procedure, yes, and so she was very successful yesterday. So now her PhD dissertation can be sent to the reviewers, and we can see here one of the youngest members of my group. So this is Julia Gabrielidi. So she is half Greek, half Polish. She was born in Athens, and now she's 2 years. She has been with us. But maybe I think that I can share this nice message with you that just on Monday, Julia she became a mother, very happy mother of a wonderful girl called Louise. Yes, so I also the other members of my group, of course. So this is the part of our institute, and here the part of our downtown, the Ratusz. So I would like to thank you very much for your attention. I would like to thank you, the audience, for staying with me till the end of this presentation. I know that it is very, very late already, and we have run for the supper, for the dinner. Yes. So once again, thank you very much. It was a great pleasure, and thank you very much for the invitation once again.
Thank you very much, Professor Bogunia. It's really very useful information, and we go out of the very, very known HLA relationships and data and whatever. And I hope it will be really useful in rheumatoid arthritis manipulation concerning especially prognosis and treatment of the disease. I don't know if it can help.
Yes, this is very important, and this we have also seen from the previous presentation that it is important that we have like a collaboration between clinician, researchers, people in the lab. Yes, because we do all these things for the patients. Yes, to find the better treatment, to find the better diagnostic markers. But this is what our work at our research laboratory is devoted to, is to find the best diagnostic and prognostic markers, yes, associated with the diagnosis of the disease, but also with prognosis, for example, of the treatment. Yeah.
How do you explain this? As I noticed at least one difference in gene incidence between the two populations, the Greek and the Polish rheumatoid arthritis patients.
But this is something very common, that the populations are different in between.
No,
Yes, and this is
in disease susceptibility, in disease. You think that?
okay. Maybe the alleles that encode this rheumatoid epitope are different, but within this sequence that is specific for the interaction with this with T-cell receptor, yes. So they are common.
Is it the same story as
So
HLA?
Yeah, but because this is because this is exactly HIA, the differences within the DRB1, yes, yes, and because we have strong linkage in between alleles within the MHC region, so therefore we can also see some differences in other loci behind this classical HLA and behind HLA DRB1. Yeah.
And I think that the other very important point will be, okay, just a moment. Another important point would be epigenetics. I think we know very well from previous studies in HLA that microbiota has to play a significant role.
Yes, of course.
I mean, microbes in the
mouth,
cavity, especially.
Yes, there are really very many different factors, and it is sometimes difficult to decide which one is the most important, yes, because as we are more immunogenetists, so we would like to find more association with the genes, but of course within our institute there are laboratories, and we collaborate with them who do the study that are associated with the microbiota analysis. Yes.
It will be interesting CCP antibodies to be also studied.
Yes.
Okay.
Yes, exactly.
It's a big discussion.
There is another question.
Thank you for your presentation. We do have single-cell RNA-seq data from patients with RA, so we might start looking into MICA now as well. Do you know what type of trigger causes the regulation of MICA in the joint? Is it hypoxia or is it TNF? I'm Achilles flutus, since you were asking. I'm a professor in University of Vienna that been working in the field of arthritis for a few years now.
Okay, so you have done some studies regarding this
We work
single
on T-cell
sequencing and
polyfunctionality
yes,
quite a lot.
functionality of MICA,
Yeah.
yes.
We haven't looked into MICA yet, but based on your data, maybe we should. And I was just wondering what type of trigger caused the regulation of MICA and if it is related to the hypoxic environment of the joint.
We can't hear you very well. Could you repeat it, please?
Right, yeah. So I was wondering if it is the hypoxic environment of the joint
Yes.
that causes the regulation of MICA.
No.
And how the different MICA alleles might change the cytokine profile of the NKs, if there's any data on that yet.
Okay, so as I saw, so there have been some functional studies that are associated with the polymorphisms that is directly translated, let's say, directly translated to the protein sequence, yes, and this valine and methionine, yes, and it can be associated, and now, as I said before, it depends on the extension of MICA expression on the cell surface. It depends on which one isoform is expressed. And then we have the differences also in this cytokine production. Because if this is valine, so we have more interferon and the cells are like, let's say, more happy to play this cytotoxic role. But when this methionine is present, then they are rather not functional. Because the receptor is downregulated.
Right, so
yes.
the only available data is on the production of interferon gamma, so we don't know about other cytokines that may be. Okay.
No, not so far.
Okay, thank you again for accepting our invitation, and we hope to continue our collaboration. Thank you.
Yeah.